All right, ladies and gentlemen. So for this presentation, what we're going to talk about are the different types of transport. So in the last presentation, we talked a lot about what basically diffusion was. Now we want to talk about some of the variations that go into transport. So the first thing I want to talk about is how there's two different types of transport. There is passive transport, which is transport that doesn't require energy. And that's really important because that means that this type of transport just happens on its own. This is the type of transport that either is the natural state of the universe or is using the natural state of the universe to its advantage. Now, passive transport obviously involves basic diffusion, like we talked about in our first presentation. But it also offers a couple of other types. So what we have here on this slide is what's called facilitated diffusion. Now this word facilitated, all right, it comes from facilitator, which is actually one who helps you do something. So facilitated diffusion is diffusion with help, all right? So what this means is that the molecules still want to spread out till they're all equally spaced out, but they can't do it through the membrane on their own. And so here you have, in our little example here, some molecules, okay? And these molecules want to get through the membrane because you can see there's a lot of them over here, which generally means there's a high concentration. And there are few of them down here, which generally means there's a low concentration. And since diffusion is all about molecules spreading out so that they all have as much room as possible, molecules tend to move from high to low. And we actually call this down the concentration gradient. So these molecules moving down the gradient want to do it. But when we have facilitated diffusion, they need help. And that help comes in the form of membrane proteins. There's still no energy, still basic diffusion. It's just that for these particular molecules to get through the membrane, they require the help of proteins. And that's why we call it facilitated diffusion. Next on our hit list is what we call active transport. So up here on this part of the diagram, you can see what we have is passive transport. So here I have molecules that are in a place of high concentration. They move to a place of low. This requires no energy, and this just happens naturally. So this includes regular diffusion. This includes facilitated diffusion. And down here we have the opposite. Here I have molecules in a place of low concentration, and here I have molecules in a place of high concentration. But instead of moving from high to low, here the molecules are moving from low to high. And so what you have here is a situation where we are basically breaking the fundamental rules of the universe. And when that happens, you must expend energy. And you can see that right here in the diagram. ATP in living things is energy. So in this situation, energy is being moved to actively transport this particular molecule from a place of low to a place of high concentration. Because energy is being expended, we call it active transport. So those are the two main types of transport. There's passive transport, where molecules move from high concentration to low concentration, and it requires no energy. And there is active transport, where molecules move from low concentration to high concentration, and that requires the expenditure of energy. Now, active transport is a really important part of the way cells work. Because cells try to set up systems so that they can constantly use diffusion as much as possible. Because they don't want to use energy if they don't have to. That's not efficient. And a great example of that is the way your lungs work. You're constantly cycling blood through your lungs so that oxygen always diffuses into your blood, because there's high concentration in your lungs, low in your blood. And carbon dioxide always diffuses out of your blood, because there's high concentration in your blood and low in your lungs. And that's why you're constantly exhaling, constantly pushing your blood through your lungs so that you can just use diffusion. That way you're not expending energy to transport the gases, you're just spending energy to move the blood. But there are plenty of situations where active transport is necessary. Chief among them is your nerves. We talked about how nerves send signals along the axon, and they do that by depolarizing the membrane. Well, depolarizing the membrane is a form of passive transport, specifically facilitated diffusion. But you need to reset the system. And if you don't reset the system, you can't send signals again. And resetting the system requires active transport. And that's just the most common example. We refer to that as the sodium and potassium pump. Now comes the most complicated type of transport, and that is osmosis. Osmosis, at its heart, is just the diffusion of water. But it's not. This is the thing about diffusion. When we're talking about diffusion, the solute is moving. So if I want to talk about how sodium ions are diffusing through water, then the ions are moving. If I want to talk about how food coloring is diffusing through water, the food coloring is moving, because those are the solutes. They're the thing I'm mixing into something. But when we talk about osmosis, we're talking about the solvent moving. So these little U tubes are, not the video site, are the way that we go about demonstrating osmosis. So what you have right here is a semi-permeable membrane, and it's the same membrane here. Now, this diagram is set up in such a way so that my solute, these little reddish dots, can't pass through the membrane. That happens. You know, chemical properties, things that are large and charged can't get through that lipid bilayer. But nature still wants the concentration equal. Nature strives for an equal concentration on either side of the membrane. No matter what, that's what nature wants to do. And that's why diffusion happens. Because if you can move the solute from one side of the membrane to the other, you will equalize concentration on both sides. But when the solute can't move, what happens is the water does. So you take a look at this diagram. You've got water here and water here. You've got water here and water here. Notice what's changed. There is still 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 dots there. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 dots there. But what's happened with these dots is that on this side, there's more water with them. Remember, concentration is the ratio between solute and solvent. So even though the solute didn't change, the amount of solvent got higher, which means that the concentration went down. So what happens is, when the solute can't move, the water does. And that's what osmosis is. Let's take a look at what this looks like in cells. So when the water moves, that has real serious implications for cells. Because your cells are not, particularly your cells, like human beings or animal cells, are not particularly strong. I mean, they're not weak, but they are breakable. And so what this does is this creates a situation like we have here in this first diagram where water can rush into the cell and the cell can pop like a balloon. And we call that the cell has lysed. You also can have a situation where the water rushes out of the cell and the cell sort of shrivels up, kind of like your fingers do when you've been in like a swimming pool or a bathtub for too long. And we call that crenation. Okay? Either of those things will kill your cells. And so it's very important that you have similar concentrations in the solution both outside and inside the cell. All right? Now, what this does is this means that we can discuss the solution outside of the cell. So if the solution outside of the cell has less solute, therefore more water concentration. So what happens is when you have a hypotonic solution, there's less solute outside. Water rushes into the cell. In an animal cell, this can cause it to lyse or burst. But in a plant cell, because it has a cell wall, it just gets turgid. Or it kind of just gets like a hose. When you have water running through a hose, you know how it's real rigid? Okay? Now, when you have an isotonic, kind of like an isosceles triangle, two sides are the same, it means that you have roughly the same concentration of water inside and outside the cell. So water moves in, water moves out, the cell is normal, it's healthy. This is what your body strives for. Okay? Same thing with plant cells, except unfortunately this doesn't give them enough pressure to stand up straight. All right? The last option is what we call a hypertonic solution, and hyper means more. And so in this case, you'll have more solute outside the cell. And so what this does is this causes water to leave the cell, and the cell shrivels up. And without enough water inside, the cell will die. In an animal cell, it just sort of shrivels and crenates. In a plant cell, because the cell wall is still there, it's still kind of attached to it, but it still shrivels and the cell wall becomes very flaccid. And so we call this plasmolysis, and that's why that says plasmolysed. And this is what it looks like when you've seen a plant that has wilted. So, that is the basics of cell transport. Obviously, we'll talk about some of this in more detail, but there you go.